Hello everybody, I am very happy for being here. Thank you for the invitation. I know that you and I connect in many ways. We connect through the science that we generate. We also connect through the passion for our daily work. But in this particular session, I want to connect with you at a different level, with the centricity that we have to put in our daily work towards our patients and the potential patients that can be treated with this compound. So I invite you to do something. If you were a patient with this chronic debilitating condition, and you know that there is a company that is trying to develop, and it is close to develop something that not only is a solution, but potentially a cure for this condition, what do you want to see in them? What do you want to hear from them? How do you want to be listened by this company? Think about it. Please realize to what extent what we do here on a daily basis can impact the work that we do for patients around the globe. And with that in mind, I invite you to keep that mindset and to discuss the topic that brings us here today without leaving that specific mindset of patient centricity. So, the purpose of the session today is to share with you the results of our phase 2 and phase 3 studies. And I also want to listen from you. What is your input on what else we can do in terms of data generation? So we will follow a 3-step process. First, I want to share with you the results of our phase 3 studies. Second, I would love to also share with you the results that my team have put together to identify which additional data is needed. What is our data gap between what we need and what we have? And finally, I have assigned some tasks for you, so you, the teams in this organization that actually generate the data, can start working in doing it. Do you have any question about the purpose or the process? Very good. So we will start with point number 1, the phase 2 and phase 3 data. In summary, as you can see on the slide here next to me, in terms of efficacy, we have an efficacious drug that has proven to be better than the existing treatments for this condition. And not only that, it has proven to be better in a way that quantifies variables that are relevant for patients. As you remember, we designed this program based on patients' needs, and we have covered them. This drug fixes problems, unmet needs that real patients in the real world have. Second, the safety analysis of it. We have a drug that has an unprecedented safety profile. Just to give you a taste of it, as you can see in this slide, the frequency of serious adverse events is as low as those adverse events are frequent in the general population without taking any drug. So basically, the use of this compound does not increase the risk of any serious adverse event. Of course, we need to take this with caution because it is only 4,000 patients. This is nothing to compare to the big clinical experience that we will have in the real world, but so far it is a very nice sign of safety. Finally, in terms of economic value, the absence of adverse events, of serious adverse events, and the positive outcomes in clinical results tell us that the economic value of the drug is positive because there will not be additional expenditures in healthcare due to the utilization of this compound. So, the second point is about what additional data is needed. And we have divided those needs in 4 areas. First, we need to prove that the drug works in every stage of the disease, either in early patients and patients that have a chronic condition. Second, we need to stratify to make sure that whatever we find in the general sample of the study, we can also see in subset of patients with different genetic backgrounds, and hopefully identify a profile of genetic signatures that respond better than others, and that would be our ideal population to be treated with this drug. Third, we need to make sure that it works in every form of the disease in terms of severity, not only in patients that have a mild disease, but also in patients that have a very severe condition. And finally, as you know, this compound treats diseases that are chronic in nature, and therefore patients can have and usually have concomitant diseases and therefore concomitant medications. So we need to make sure that it works in every population taking any drug and that there is no interaction with it. So this takes us to our third topic of the process today, that is the data generation plan. So as you can see on the table next to me, I have assigned this data gaps to the different functions in your teams, and in the following days, my team will reach out to you to organize workshops so we can go in depth to find ways to generate the data that is missing through innovative experimental models. Do you have any questions so far? Well, thank you very much. This has been a very useful session. I really appreciate your time and your invitation. And as Mike mentioned just as the beginning of the meeting, and Mike, I think you're absolutely right. As long as we keep patients' well-being in mind, we're going to be doing a task that is going to be useful not only for science, but the most important thing, for people affected with this drug and their well-being. So thank you very much for your time. I'm looking forward to speak with you again.